Welcome you all to my channel Omni Bio Entrance, and today we will be discussing IIT JAM Biotechnology question paper conducted in the year 2013. I will be discussing only the biology part. This is part 2 of the discussion. If you are new to my channel, please do like, share and subscribe. I have uploaded still previous year question papers of IIT JAM Biotechnology. You can refer them in my channel playlist. Coming to the first question. For a single enzyme single substrate reaction, the half-life of an enzyme can be calculated using the following expression, where K is the first order rate constant or deactivation constant. So for a first order reaction, K is equal to 2.303 by T log C by Ct. Here T is T half, they have asked for half-life. Ct is CO divided by 2, that is concentration after time t, and CO is concentration at the initial time. K is equal to, substitute the values, you will get as T half is 2.303 K by log 2. Answer is option B. Then next one. The transmittance of a solution containing 10 raised to minus 5 molar ATP is 0.75 or 75% at 260 nanometers in a 1 centimeter path length cuvette. According to Beer-Lambert law, the absorbance is, so T is 0.75 at 260 nanometers, and according to Beer-Lambert law, absorbance is equal to log I0 by I, or A is equal to minus log I by I0, and transmittance is equal to I by I0, so that is equal to absorbance is equal to minus log T, that is equal to minus log 0.75, or you will get the answer as 0.125. Answer is option C. Then next one. Which one of the following is applicable to an enzyme catalyzed reaction that follows zero order kinetics as per Michaelis-Menten model? So the answer here is option D, S greater than, very much greater than Km, and V is equal to Vmax. Then next one. The free energy change under standard conditions upon hydrolysis of a high energy bond is large and negative. ATP, phosphocreatine, phosphoenol pyruvate and acetyl coenzyme A are examples of molecules that contain such high energy bond. Which of the following are high energy bonds present in these molecules? N, P, CS, PO. So all three of them are found in these molecules given above. So the answer here is option D, that is P, Q and R. The next one. Secretory IgA can exist in a protease-rich mucosal environment because the secretory component is secretory component masks the site susceptible to proteases. The secretory component is a protein that binds to the Fab region of IgA, masking its site from proteases. Then next one. Suppose the surface area of an erythrocyte is 167 micrometer square. Also suppose that the membrane of this erythrocyte contains 4.15 into 10 raised to minus 16 mole of phospholipid and 3.1 into 10 raised to minus 16 mole of cholesterol. The cross-sectional area of a phospholipid molecule is 0.7 nanometer square and that of cholesterol molecule is 0.38. Phospholipid and cholesterol isolated from this membrane were spread into a monolayer. Okay. The ratio of the area of this monolayer to that of the erythrocyte, to that of the surface area of the erythrocyte. So to calculate the total area of the monolayer, you need to multiply the number of moles of each by their respective cross-sectional area. So phospholipid area will be 4.15 into 10 raised to minus 16 into 0.7 moles. Sorry, 0.7 nanometer square. So that will give you 2.905 into 10 raised to 16. Similarly, cholesterol area also you can calculate and find out. Then you add these, you will get the area of the monolayer. Then the required ratio will be total area of monolayer by surface area of erythrocyte. That will be 2. Answer will be option D. Then next one. For the Na plus mediated active transport of a molecule of glutamic acid from a concentration of 0.1 millimolar outside the cell to 20 millimolar inside the cell, calculate the minimum number of Na plus ions required to supply the necessary free energy. T is 37 degrees, pH is 7, delta psi is minus 70 millivolts, and Faraday is constant is 23062 calories per mole per volt. Delta G released for the transport of 1 Na plus is 3.3 kilocalories per mole. Glutamic acid carries a net charge of minus 1 at pH 7. So from the Nernst equation, you have the formula E is equal to E0 minus 2.303 RT by NF log Na plus inside and by Na plus outside. So F they have given, E is your this one delta psi, that is minus 70 millivolt, E0 is 0, R is 2 calories per mole, concentration of Na plus inside is 20 millivolt and outside is 0.1. T is 37 degrees Celsius. Convert it into Kelvin. 37 plus 273, that is 310 Kelvin. Substitute the values and you will get N is equal to 2. N is what they have asked. So answer here is option C. Then next one. NAD and NADP are two important cellular electron carriers. NAD is generally associated with catabolic reactions and NADP with anabolic reactions. A cell will have a fixed number of NAD molecules and a fixed number of NADP molecules. In a normal cell, most of the NAD and NADP molecule will be reduced state. No. NAD molecule will be in the reduced state. No, both will be in the oxidized state. Answer is option D. NAD and NADP molecules will be in the oxidized state. Now next one. In the pedigree of a family is affected by a certain genetic disorder is shown below. Microsatellite markers from these individuals were PCR amplified, separated by gel electrophoresis and detected by two probes P1 and P2. Probe P1 is marker for M1 and probe P2 is marker for M2. The resulting banding pattern are shown along with the pedigree diagram. From this data, it can be concluded that now, if you see here, this is the father and this is the mother. The father is deceased, so he is having these kind of bands for P1 and P2, whereas mother is not deceased, so P1 and P2 show this type of band. But the offsprings you see, they get this band from the father and this band from the mother. And for P1. For P2, they get this band from the mother and this band from the father. Similarly, here also they get this person gets this band from the father and this band from the mother. Mother is having only one P1 band. And in P2, if you see, she will get this band from the father and this band from the mother, whereas the normal progeny is having entirely different banding pattern. But if you see the banding pattern of this deceased progeny, you will see that P1 is very much similar in both the deceased progeny bands in P1. This band, this band, this band and this band. Whereas in P2 the bands are entirely different, whereas the normal progeny P1 is will be obtained from the mother, but the band obtained from the father is entirely different. This band is obtained from the father, so the person is not deceased. So you can conclude that the trait is linked only to the marker M1 because P1 is the marker for M1. So the trait is linked only to the marker M1. Answer is option C. Then next one. Which of the following statement is not correct? SDS page separate protein based on molecular weight. That's correct. Gel filtration chromatography can be used to determine the molecular weight of a protein. Yes. Ion exchange chromatography separate proteins based on their size, not on their size but based on their net charge. So C is not correct and that is your answer. They have asked for the one which is not correct. Membrane dialysis is commonly used for desalting. That's also correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, please subscribe to it. Then I have uploaded still previous year question papers of IIT JAM Biotechnology. You can refer to my channel playlist for the same. Thank you.